Hey gang, this is your video for section 3 of chapter 11, chapter 11 on cell communication, and the first picture we're looking at here is the big picture of basically how a signal gets from outside of a cell, gets received, and gets to the inside of a cell where it needs to be. And so what we talked about last video was the reception of a signal, and that reception basically, when that process, step number 1 here, is done, you're going to have gotten a signal that was received by a cell, and somehow that signal is now on the inside of a cell in one form or another. So typically that ended up with some sort of activated enzyme on the inside of a cell was the end of reception, and that was the signal, an activated enzyme on the inside of a cell. So now what we're going to pick up with is the transduction step, which is taking that signal that is just on the inside surface of the plasma membrane or has just gotten inside of the cell, taking that signal and somehow carrying it, maybe making it a larger or more common signal, but somehow carrying it to wherever the response needs to happen. Oftentimes it's the nucleus because we're turning on a gene, or maybe it's turning on some kind of proteins that will help a cell move, or turning on enzymes to make some kind of a protein. Anyway, so we're going to focus in on the transduction part of things. A term that is commonly used here is a phosphorylation cascade. And this is one type of transduction, is a phosphorylation cascade. It ends up being very, very common, but there are other types. We'll go through a couple other types as well, but just keep in mind that whatever the book is showing you here, it's honestly the tip of the iceberg as far as how transduction could happen, and this is just happens to be the most common. So anyway, this term cascade, I wanted to give you a visual on that. When they talk about a cascade, typically we're talking about a waterfall, if you were using it in common language. So here's a waterfall, and you probably wouldn't use cascade to describe like Niagara Falls, where it's just one huge drop, where you're at the very top of a waterfall and the water just boom straight down. It's going to be more like this one if we're using the term cascade, where there's the water's dropping from one level to the next, to the next, to the next, and it's hitting a rock and then bouncing to the next rock down and the next rock down and the next rock. That's a cascade, and this is kind of how if we draw out this type of transduction pathway, it's kind of like that, where in this case it's activated molecules are kind of bouncing to the next activated molecule, bouncing to the next one, and the next one, and the next one, and so on. So anyway, another thing before I even get into the diagram here, so that's that term cascade. The next one is something called a protein kinase. And a protein kinase, first of all, a kinase is an enzyme that phosphorylates something, typically taking phosphate from an ATP and putting it onto something else. I defined that in the last video, I believe. And then a protein kinase is just it adds a phosphate to a protein. Okay, so we're going to use a lot of those in this, and these are very, very common in the cell to have these protein kinases. Then another term that shows up here is a relay molecule. Technically, all of these would be called relay molecules. They're carrying the signal from one place to another, or from one molecule to another. So where we typically would have left off after our first step, our reception, is we would have some form of a relay molecule made, whether it was an enzyme that was turned on or something else, but there'd be a relay molecule. And our transduction pathway is going to start when that relay molecule here basically joins an inactivated protein kinase. So it's just a protein kinase that is not functional. It's an enzyme, and it's not working. So when that relay molecule and this inactive protein kinase, when they join together, it activates the protein kinase so that it can do its job, and its job would be to put a phosphate on another protein. So then we have protein kinase 2 coming into the picture. Once we've activated protein kinase 1, if it can find a protein kinase 2, then it can activate it. The way it activates it is it joins it, that would be its substrate, and it will find an ATP, take a phosphate off the ATP, and put it onto protein kinase 2. Okay. Well, now we've activated protein kinase 2. It can do the same thing to another protein kinase. So then you'd have protein kinase 3 going through the exact same process. Protein kinase 3 going through the same process, and this could go on for a whole lot of steps. But eventually we're going to get to a protein kinase that its job is to phosphorylate some protein, usually an enzyme, but some protein that is required to start our cellular response, to actually do what we're supposed to do. Okay, then we have our active protein, and that'll go into our step 3, the cellular response. So this really isn't like a huge deal. It's kind of whisper down the lane type thing, where we're activating one, and it goes to the next one, activates that, goes to the next one, activates that. It's going to keep going until it gets to the goal, whatever that is, to turn on some sort of a protein or activate some kind of a protein. The big question, and I'm actually going to talk about this at the end of I think video 4, section 4. The big question is why this seems so complex. Like why would we have this whisper down the lane type thing when we could just activate protein kinase 1 and then have that go and activate the initial or the final protein for the cellular response. And we'll get to that. There's a bunch of reasons for that. But yeah, just kind of quick overview on those is we can have more control over this process. The more steps there are, then the more places that we could control this. Other things are we'll find that if we had one protein kinase 1, it has the ability to activate more than one protein kinase 2. So we can end up having large quantities until we get down through a bunch of steps. We've amplified, they call it, but we've made a whole lot of the protein kinase 3, which means we can activate a whole lot of the protein that needs to be activated for the cellular response. So that's one type, this phosphorylation cascade. It's a good picture of a transduction pathway. Now we'll go through some other concepts that come into these transduction pathways.